So our next speaker is going to give the introductory talk, and he's a clinical oncologist who surprisingly has been working for the last 15 years with theoretical physicists, trying to understand drug resistance in cancer, cancer growth, and various other issues in cancer research and actually curing or treating real patients. And I've been working with him for the last 6-7 years, and it is, I was amazed that actually clinical oncologists can try to understand biophysics, kinetic equations, and so forth. But he's been exposed to theoretical physics, and he actually understands the new ideas and the different way of thinking that physicists bring to the field. And for me, it was very important to have someone who has actually comes from the other side of biophysics, or let's say of biomedical research, to talk to you about the importance of bringing quantitative methods and quantitative models in really clinical research. So from a very practical point of view, your research will be very important all the way to medicine and treating patients. So I'm very happy to introduce you to Fojo. He's been at NIH, at the National Cancer Institute, since 1983 until 2015, where he's moving to Columbia University in the next few weeks. Thank you, Tito.
Okay. Okay, Krastan, I'm not going to give that talk. So, actually, I asked Krastan what I should talk about, and it was incredibly vague. So then I looked at all the presentations you all were having, and I said, boy, this is a really diverse audience. And what could they do, and or what could they be interested in? And so I also thought, well, it's early in the morning. They don't want to get into anything very, very deep. I want to let them in gradually. So hopefully we enjoy it, or at least learn something. Just put date on file. But there's good. So, but thank you for the introduction. I actually got involved in this by, many more years ago than we want to admit. Krastan came to talk to me, and then I realized how much a physicist and a medical oncologist had in common. And so maybe that's the theme of this presentation. So does anybody know what this is? Other than a pretty flower, right? It's actually, you might ask for a periwinkle. And who would have thought that this drug would actually be incredibly important in the therapy of cancer? Actually, there's 2 drugs that have been extracted from the periwinkle. One is vinblastine, and the other is vincristine. And you can see the structure here. And this actually sets the pattern for structures that we extract from nature. They're incredibly complex. And so, at many levels, incredibly interesting. So vincristine was first given to children in the late 1950s and was eventually approved by the FDA in July 13 as a drug called Acute. The drug was initially discovered by a team led by Dr. J. Armstrong, and it was marketed by Eli Lilly and Company. At that point, making drugs for cancer was unheard of. So this was one of the first drugs to be made for cancer. And when it was given to children, it was initially given as a single agent. In the early 1960s, it was added to other agents, and for the first time, children went into a prolonged remission. So the history of leukemia, basically, in 1946, a fellow by the name of Sidney Farber, after whom the Farber is named in Boston, used antifolate agents to treat leukemia in children. It was kind of a sweet and sour sort of story. They would have great responses, and then shortly thereafter, after the disease had appeared to go into remission, they would have rapid recurrences, and they would die. In the 1960s, the addition of vincristine and steroids led to marked increase in the response rate and eventually cures, so that survival rates increased to over 90%. Today they're over 50%. Today they're over 90%. And then so we started to realize that leukemia was complex and began to make even more inroads into it. So how do vincristine and vinblastine act? They act by affecting the microtubules. And the microtubules, nearly all of you I'm sure have studied, as important structures in mitosis. But they're equally, if not more important, as the railroad on which everything traffics in cells. So if you want to get from the membrane to the nucleus, and from the nucleus to the membrane, you jump on a protein of one of two families, the denane or kinesin families, and you traffic on microtubules in an outward direction or in an inward direction. And I remember when I was in high school, you have the cell, a nucleus, and maybe two or three things. You thought anything could swim in the cytoplasm. Obviously, the cytoplasm is packed, it's jam-packed. So if you want to get from one place to another, you can do it, but it's faster if you jump on a microtubule. What vincristine and vinblastine did was that they led to depolymerization of the microtubules. And in fact, if you take a cancer cell and you treat it with vincristine and vinblastine, you come and you look back and you look at it, the three-dimensional structure that is the microtubular network, that is this cytoskeletal network, has completely disappeared. It's been dissolved. So these were interesting drugs, and it sort of made sense that maybe by interfering with microtubules, we were interfering with mitosis. And this, on the lower right-hand corner, you can see how beautiful a microtubule network can look. And as I say, when you add vincristine or vinblastine, this totally disappears. And likewise, what you see then is also that cells, as they attempt to go through mitosis, halfway in mitosis, they are arrested because this network that is supposed to modulate cell division is destroyed. So enter this fellow, Monroe Wall, and then this fellow, Mani Wani. And Wall was a botanist, Wani was a chemist, and in the 1960s, in the late 1950s and the early 1960s, the NCI decided that it had to basically mine natural products to try and find chemotherapeutic agents. They had seen that a drug like vincristine and vinblastine had come from the periwinkle, and they figured there must be a lot more products out there that we have to mine. And Wall had mined a lot of products, and he needed someone like Mani, a chemist, to take them forward. So what were Mani and Wall doing at that time? They had thousands and thousands of extracts, and they were looking for them to see what were active properties and which might have anti-cancer properties. So does anybody recognize this tree? If you're from the Pacific Northwest, this is the yew tree. Here's another flavor of it. They're incredibly beautiful, incredibly large, and incredibly endangered. So this is the Pacific yew tree, and it's called Taxus brevifolia, which is why the drug that I'll discuss now, which is called Taxol, was developed from this tree. This is Taxol, and a woman by the name of Susan Horowitz that I'll talk about in a few minutes. When she looked at the structure, she said, only Mother Nature could have come up with something this complex. No human being would have ever thought of this. And we know an enormous amount about Taxol, but as I'll tell you at the end, we still also know very little about Taxol. Taxol was eventually developed into a drug, and it was sold by Bristol Myers Squibb, and I'll tell you a little bit more about that in a second. So a fellow by the name of Jonathan Hartwell had been sort of given the task of developing the natural products. And he convinced Wall to send to the NCI these ethanolic plant extracts that he had. And then, Wani, who was working at that time in Research Triangle, said that the Institute, which was called RTI, that the Institute was nothing but four walls. It was not until the fifth wall arrived that the chemistry programs in the form of natural products laboratories started moving. And Wani described Wall as a go-getter and a very dedicated scientist. About Wall, Wani adds, persistence was his greatest virtue, but patience was not. And I've only met Wani, not Wall, and Wani is, and this is not meant in any, is your typical, very patient, very good Indian scientist. And I suspect only someone like him could have put up with Wall. But what was apparently oil and water worked out really well, and they had 40 years of a very fruitful collaboration. So the crude extract showed potential for activity for becoming candidates for fractionation. They went to the NCI. Wall had been in charge of a program at the Eastern Regional Laboratory, again, harvesting all these natural products. 1,000 extracts of plants were sampled and they were sent. And the first one that actually Wall and Manny worked on was something from a plant called Campathica acuminata. That eventually became a chemotherapeutic agent called camptothecin. And I'll come back to that later. And so then Wall and Manny began to work on these many numerous compounds. But then entered this wonderful woman, this is Susan Horowitz. And Susan was at Einstein, and the NCI contacted her and said, we've got this really interesting compound, would you be interested in looking at it? So they had collected various ones, and as Susan Horowitz says, as the story goes, Taxol was collected during the summer by botanists and some college students that were out west to California and Oregon. Actually, the first parts of the yew tree were collected at the end of August, just before the college students were coming back. And it was that Wall took back to the laboratory. And then Dr. Shepard says that testing in vitro initially showed that it was a potential interest, particularly the bark. So this will become a problem, as you'll see in a second, but a problem that was eventually solved. So Susan says, I received a letter from the NCI asking me if I would look at this drug and try to determine its mechanism of action. It wasn't until I saw the structure of Taxol that I wrote back to them and said, this is a compound that I think would be interesting because the structure of the molecule was unusual and unique, and that was true. So she says, I had a new graduate student who was looking for something to do for his PhD thesis. It was Peter Schiff. And we looked at this structure and I said to Peter, okay, this is the deal, 1 month. You can work on this drug for 1 month, and if you think it's interesting, we'll go on. But after 1 month, we can't move on it. We've got to go to the next thesis project. Well, by the end of the month, we knew that it was interesting. And Peter Schiff and Susan Horowitz are actually instrumental in the development of Taxol. So what had they done? They had added Taxol to HeLa cells, and then we had stained those cells to look for microtubules. They were trying to find this mechanism of action. The amazing thing was that when we looked under the microscope, these cells were just loaded, filled with microtubules. And that was a very unusual thing. I mean, Taxol clearly was a drug that blocked cell mitosis. We learned that very quickly. It was very cytotoxic. We could use it at very low concentrations and kill cells. So, to me, this is interesting. Because as it turned out, what Taxol did was that unlike vincristine and vinblastine, which depolymerized microtubules, Taxol actually hyper-stabilized microtubules. So that you ended up with these rods of microtubules themselves that were just like the disassembled microtubules, non-functional. But I often think, if you had been in a room with a bunch of smart people, and you said, well, we got to figure out a new drug for cancer, what do we have? And somebody said, well, vincristine. Oh yeah, what does that do? Well, it, disassembles microtubules. I don't think many people would have thought, why don't we make a drug that assembles microtubules as a way to treat cancer? Let's just go to the total opposite end of the spectrum. But in fact, that's what Mother Nature had figured out. Actually, what Mother Nature had figured out was that microtubules were a superb target, and any way that it could get at it, it would get at it. There were other drugs, says Susan, like the vinca alkaloids, that could have done that, but when we looked at those cells and saw this massive microtubules, this was unique and totally unexpected. So, Bruce Chapman, who at that time was at the NCI, he's been at Harvard for the last decade or so, noted that the compound was very insoluble. And what I'm going to show you here is the challenges of developing drugs. Today, we think it's easy, but it really isn't. So it had to be dissolved in cremophore, which is a lipid emulsion. And in that form, the cremophore caused hypersensitivity responses. In fact, when the drug finally was given to patients, it was nearly terminated after the first 3 patients. All 3 had major anaphylactic reactions. They nearly died. Their blood pressures tanked. They had difficulty breathing. Everybody thought they were going to die. What was this? Well, it turned out it wasn't Taxol, but it was the cremophore that it was dissolved in. And it took a couple of very clever and patient clinicians to work this out, Peter Warnick among them. And at that point, then it became apparent that the drug had activity in ovarian cancer, and then everybody got really interested in it. So now you had a drug that was very unique, had activity in ovarian cancer, and its development problems, at least to some extent, had been worked out. Mike Krieger at that time was also at the NCI. But it was unfortunate, he says, that the supply of Taxol was really, truly at a critical level. And I was at the NCI at that point. We actually had a SAR of Taxol. Basically, a guy who's still there, who worked with me, whose job was to keep tabs of the number of vials of Taxol that we had. And the mantra at that time was that you got 2 treatments, 2 cycles of Taxol, and unless you had a great response, you got no further Taxol, because there wasn't enough to go around. And Wyndham, as the fellow saying, every Friday had a meeting with everybody, and how many vials of Taxol do we have left? And they were having a lot of difficulty producing it. Now, there were so many people interested in it. We would get phone calls from loggers in Oregon saying they had this tree and they were wondering if it could be of help to us. So there was a tremendous interest in trying to do whatever they could to help us develop the drug further. And it was really appreciated, but there was another problem, and that was this. Who knows who these guys are? So this is Dennis babies. There's a spotted owl. So the spotted owl was endangered. And the spotted owl lives in the yew tree. And the problem was that Taxol came from the bark of the yew tree. And if you removed the bark from the yew tree died. And not only was the yew tree endangered, but more importantly, the spotted owl was endangered, and the yew tree was its natural habitat. So this had become a real big problem. Where were we going to get Taxol? And it was so complicated, no chemist thought that they could synthesize this de novo. And there were many studies. Medicinal tree used in chemotherapy drug faces extinction. You can see here, they're taking the bark off the tree. And that was the only way to harvest Taxol. So the medicinal tree, so all of this, so Taxol was discovered by the National Cancer Institute. All 11 species of yew were found to contain Taxol. But we had to get it from the bark of the yew tree. But then a breakthrough came. It turned out that it was possible to extract Taxol from clippings, and actually that's not true. What was possible to extract was a precursor for Taxol. And some 6 to 8 steps later, smart chemists could make Taxol. And so all of a sudden, we could have it from a renewable resource, the clippings of the tree, and so consequently, the Taxol problem was solved. And this new source for cancer drug found, medicine researchers say they can now make more Taxol, make Taxol from the leaves and branches of yew tree instead of the bark. The finding means forests at homes with a spotted owl would not be devastated. So at the end of the day, this little owl actually led to the development of a new method that protected the yew tree. Bristol-Myers Squibb constructed a Taxol production facility, and before long, we actually didn't have a shortage of Taxol. We actually had an excess of Taxol. The new process will end on dependence on the Pacific yew tree and the use of the bark from it should decline pretty sharply. The spotted owl is safe. So, as Mike Krieger noted, this was a collaboration with the pharmaceutical industry, and we were able to identify a more rapid way of making Taxol available. We also found that Taxol could be derived from a renewable resource, and therefore we didn't need to destroy the yew tree. Now, what Susan says, and this is true, and I think it applies to all of you, so I think all of you should end up working on cancer research, okay? I mean, that's what I think is important, you know? So just find your way there, okay? There's many ways to get there. But as Susan says, the Taxol story was a multidisciplinary story. People like myself, lab people, biochemists, pharmacologists, people in the clinic, oncologists who worked on it, people like Monroe Wall in medicinal chemistry who isolated determined the structure, and of course industry, Bristol-Myers Squibb, were involved in development of the drug for patients. And then something interesting happened that's even in many ways more important about Taxol. When Taxol was developed, drug companies simply were not developing drugs for patients with cancer. It was generally thought a patient with cancer takes a drug for a couple of months at best, they die. Who wants to develop a drug for someone who's going to take it for 4 weeks, I'd rather develop an antihypertensive that someone's going to take 40 years, insulin that they'll take 40 years, you know, a lipid-controlling drug that they'll take for 50 or 60 years. Those are far more attractive. As Susan said, I mean, there was no indication that this would be profitable. In fact, it was Taxol was actually the first $1 billion drug in cancer. And all of a sudden, industry got interested in cancer. And beginning in the mid-1990s, the industry then really got into cancer in a big way and changed the development of drugs. How did that change? Have you ever seen this? This is what we call the quinome, right? So there's a bunch of tyrosine kinases, and there's also serine kinases in the cell. It's about 700 of them. And what they do is they catalyze any number of reactions. The majority of them, as regards cancer, and even not as regards cancer, are surface proteins that are signaling proteins that basically have part of the protein on the outside, they receive the signal, and then they transmit it through the membrane. And on the inside is the portion that then phosphorylates tyrosine on proteins that are their substrates. So the protein kinase complement of the human genome was eventually solved, and there were over 700 kinases. Actually, in the 1980s, it had been predicted that it would be about 1,000, so it wasn't so far off. What that led then to the development of what we now call TKIs, which are tyrosine kinase inhibitors. And there's about 15 of them approved by the FDA. So where did this TKI business start? So it started with a company called Sugen. And Sugen came to life in the early to mid-1990s. And Sugen was the brainchild of Josie Schlesinger and Axel Ulrich. And not that these guys have big egos, but that's why the S for Sugen and the U for Ulrich. Schlesinger and Ulrich, and it was to be a genetic company. So they founded Sugen. And actually, I went to Sugen in the 1990s, in the mid-1990s. It was a wonderful company, full of really smart people who suffered from only one thing that you should never suffer from, arrogance. Okay? They knew that they were going to cure cancer. They were just so sure that they were going to cure cancer because they were going to develop these TKIs, these tyrosine kinase inhibitors, that would inhibit the critical tyrosine kinases. But it was just a great place. I mean, it was the future of cancer medicine. And they were developing basically compounds that were going to fit into the pockets where ATP would fit. And they would block the activity of all these tyrosine kinases. And there was a lot of modeling going on. Now we had physicists. We had a lot of people with X-ray crystallography giving us really critical understanding of what these pockets look like. And so Sugen was flying high, but it eventually failed, and its remaining assets were sold off before the decade was over. But Sugen actually had launched a new concept, and actually, there's no doubt that they were pioneers in the field. I guess they were just good scientists, they were just poor businessmen. And so the company failed. But that led then to what was probably the perfect storm. And what you need for this is a really nice, smart guy, and that's Brian Drucker up in the right-hand corner. And actually, I was his chief resident back in 1981, and I know him to be a really smart and equally nice guy. And he was working on a disease which is called chronic myeloid leukemia, or CML. And it was known from work in the 1970s that CML had a gene rearranged that was called BCR-ABL, where the Abelson kinase was captured by another gene, BCR, and this hybrid molecule then was thought to be critical for the genesis and the maintenance of CML. And Brian and other people, but Brian actually was the pioneer of this, felt that if they could identify an inhibitor for this tyrosine kinase, the Abelson tyrosine kinase, that maybe CML could be cured. And that became a drug that at that time was initially called STI 388 or 188 or whatever it was. It eventually became imatinib, and then was eventually sold as Gleevec. And then this was all the rage in 2000 when it was first presented. There's new ammunition in the war against cancer, and these are the bullets. That's actually pictures of imatinib at that point. The original study was published in 2001 in the New England Journal. Now in cancer, you usually like to have overall survival, but this was so exciting. No overall survival was presented here, but everybody was having dramatic responses. You can see here that in a phase 1 study where they were looking to see what will be the dose that patients tolerate, even at the very low starting dose, 33% of the patients had a response to this treatment. By the time they got to a dose of 300 to 1,000, 100% of the patients had a response. 98% of them were complete response. These were patients in whom the leukemia had disappeared from the blood, and then the majority of them, when you went looking for it in the bone marrow, could not be found either. A 5-year follow-up presented later showed you the survival of these patients. It was remarkable, okay? These are patients, CML-related deaths. These are all deaths. The reason is that patients who get CML are usually elderly patients, and they'll die of other things. So actually, more of them were dying of natural causes, or things other than their leukemia, than were dying of CML. Now there is a problem. CML is a chronic disease. You never hear about chronic pancreatic cancer, chronic colon cancer, chronic breast cancer. So CML at some level was a good but an easier model to begin with. It doesn't undervalue the value of Gleevec and the treatment of CML. But there was another type of leukemia, and that was acute myoclastic leukemia. And CML would often progress to an accelerated phase where the patients, instead of living 5 years or so, lived only 6 to 9 months, or eventually go into blast crisis where they lived 3 to 6 months. And why this happened, we thought that it was because not only did you have expression of PCR-ABL, but the cell acquired other things that made it not just a cancer cell, but a rapidly growing and very malignant cancer cell. And it included, among other things, activation of beta-catenin. Actually, at the time that Brian published that study in the New England Journal, there was a second article that accompanied it that has been cited about 100 times as much. And it was in patients with blast crisis and accelerated phase of CML. And you can see here the number with a response, 11% to 20%, a far more modest number than the 100% that we have seen in CML. This highlighted for us that in these patients, it was harder. And in these patients, actually, you could look at their relapse in those who responded. So now we're looking only at the 11 to 20% that responded, and they quickly relapsed. So that CML was treatable, but the more accelerated, more aggressive phases were not. And in fact, today, this is actually from 2013, but if you look at 2015, it's still so. There is something called the National Comprehensive Cancer Network, where you can go and look up what should I do next with this patient. And if you see the advanced phases, the accelerated phases, or the blast crisis of CML, still we're trying to find an answer to this cancer. And the suggestion is that patients enroll in clinical trials, clinical trials, clinical trials. Now, one thing about Gleevec was that it was active not only against the Abelson kinase, but it was also active against something called c-KIT, which actually was found to be the kinase that promoted the growth of a tumor known as GIST, for gastrointestinal stromal tumor. And here you see a patient with GIST. And what this is a fluorodeoxyglucose PET scan. And what the patient has been told to do is don't drink anything overnight, don't have any sugar, don't have anything, not even a little elements, and come 6 hours after you've been like this to the nuclear medicine department, and we'll inject in your veins some glucose that is labeled with fluorine-18. And then what we'll do is we'll scan you in front of a machine that detects radioactivity. And you see all of those little spots are tumors. and they're very avidly taking up glucose because tumors utilize glucose only less so than this structure up here, which you all are currently utilizing, and it's on fire because it's using glucose. But only the brain utilizes glucose like tumors do. And this patient had GIST, was treated with imatinib, and here's the patient. Actually, this is a patient who was treated for a few months, but very quickly the tumors disappeared. It turned out that in addition to being a very good inhibitor of BCR-ABL, or the adenosine kinase, imatinib was also an inhibitor of c-KIT. Eventually, this drug was given to patients with newly diagnosed GIST, and here you see the survival, and this is 4 years later, of patients who were getting what we call adjuvant imatinib. After they had the diagnosis made, they began on the drug. And actually, they got it for a short period. Now patients start imatinib and never come off of it, and they're alive 10 years later without a recurrence of their disease. What we really had discovered was that these signaling pathways that we thought were so simple, as we had thought maybe in CML, were actually very much more complex. And this is work from the group at Johns Hopkins. And basically, what we found that, for example, in pancreatic cancer, this is an engine that runs not just on one cylinder, but on 12 cylinders. And if you block one, or even two, you're unlikely to have much of an impact. Meaning that some of these cancers are a little more complicated than we had hoped they would be. So, but what about Sugen? Actually, what happened was that Sugen had a fire sale and sold off all of its acids. And what had happened was the scientists at Sugen, who began Sugen, said, we're going to have these magic bullets, and we're going to develop drugs that target the tumor and spare the body. Up until that point, nobody wanted chemotherapy because it's so bad. If you hadn't had it, you knew somebody who had had it, or you knew someone who knew someone who had had it, and you'd heard about how bad this was. So they were going to take these drugs and develop them to be critical and to target only one thing, and that was the kinase that was present in these tumors that presumably was really important in the growth of the tumors. Well, for one thing, that was flawed reasoning because tumors don't invent kinases. They just usurp naturally occurring kinases. And so not only are they in tumors, they're in every normal tissue in the body, and in some at very high levels. So there were a lot of toxicities that came along with many of these drugs. And then there came to be what we call off-target effects. And that is that these drugs have nearly all been developed to target the ATP binding domain. In fact, when Ulrich and Schlesinger began Sugen, everybody said, you're out of your mind. You'll never be able to develop a drug that's specific enough for one kinase because they're all using ATP. But Ulrich and Schlesinger were convinced that the binding pocket would be sufficiently different to put in a drug that blocked only one and only one binding pocket. Well, they were only partly right. You can develop drugs that block a few kinases, but there is no drug that blocks only one kinase. In fact, the most specific one still remains Gleevec, and we know that it blocks the adenosine kinase, c-KIT, and then one other kinase at somewhat levels. But the majority of kinase inhibitors were blocking multiple sites because everybody is using ATP. And so they had this idea, and so what they were doing was they were screening the TKIs that they had. And if it seemed to have a lot of activity against a lot of other kinases, they weren't interested in them. Because rightly or wrongly, they thought these will be very toxic. Okay? And they called that off-target effect, because you were aiming for one kinase, but you were inhibiting a bunch of kinases. And so Sugen had one compound. Unless you think that they're, bravado ended at the name of the company, they had actually named this compound sunitinib. Okay, again for Schlesinger and Ulrich. And they were really excited about sunitinib until they profiled it. And sunitinib had a bunch of off-target effects. So, although Sugen never produced an FDA-approved cancer drug, as I told you, company pioneered the field of tyrosine kinase inhibitors. And Sugen scientists had a depth of knowledge that was exceptional, and there's no doubt about that. So, excuse me, Pfizer bought off the remnants of it, and here you have the genome. And what we're showing here is which kinases are inhibited by these compounds that we're producing. In imatinib, you can see that out of the 700 kinases that are being tested here, has activity against a very small number of tyrosine kinases. And by the way, this shows you the amount of drug that is required to inhibit. So it's very effective at inhibiting a very few kinases. For other kinases that might inhibit it, it requires 1 to 10 micromolar, and that's a very large concentration of drug that you're unlikely to ever achieve in a patient. But look at sunitinib. It was inhibiting every kinase you could test. So the people at Sugen had decided they weren't interested in it, although it bore their initials. And it was practically the only drug with any hope that they sold to Pfizer. Here you can see a bunch of other drugs. We got better. These drugs are approved, gefitinib, erlotinib, lapatinib. And as you can see, they have very little effect on the majority of kinases, or very high concentrations are required. And it's comparable to imatinib. But they also are other drugs that were approved that had a lot of activity. And that's because of this fellow, Jean-Pierre Armand, a very wily French physician who had developed Myoclastic camptothecin, a drug that initially was so toxic that it was felt it would never make it in the marketplace, but it did. And that's because Jean-Pierre Armand wasn't deterred by toxic drugs. So sunitinib was really toxic. And he said to Pfizer, that's okay, I'll do it. And it was really toxic. And I remember being at the meeting where he first presented the data, and he went in and he said, 40% of kidney cancer patients are having marked responses to this drug. And he said in his own understated way, it's really toxic, but we'll figure this out. And eventually he did, and the drug was marketed. It was marketed as sunitinib initially. And in renal cell carcinoma, 47 to 44% of the patients had responses. Their disease was not progressing as quickly as patients who weren't getting it, and the overall survival was longer. Now, so to end, these are just some vignettes. They're mostly successful vignettes, but there's a lot that aren't successful. And why I think you all need to get interested in cancer is because despite the advances, we still have over 550,000 Americans that die of cancer every year. So there's a lot of room for improvement. If we leave it only to oncologists, or only to biologists, or only to, a fraction of the scientific world, we're not going to be able to make progress. So as you go along, realize that you can make a lot of contributions in ways that you don't understand. And that, cancer drug development is a multidisciplinary thing. So you don't all have to work in cancer, okay? Just half of you, all right? So thank you very much, and I hope that this was something like what Creston had in mind. Thank you. No, I'm not.
So, okay, there are new drugs, but the targets are pretty much microtubules or tyrosine kinases, or there's any other complete different targets that people are thinking about these days?
Yeah,
Can you comment on?
people think about targets, but I mean, anybody who works on a protein ends up thinking that's a target, you know. And the majority of targets end up not being targets. But we've gone beyond tyrosine kinases, and there's an increasing number of targets. And so I think, you know, we're going to make progress in the future. And it won't be on, I think we've had the wave of tyrosine kinases. We probably will see a few more come into the clinic, but the majority of them, I think, have been exploited aggressively. So until we find that a certain tyrosine kinase is really important in a subset of tumors, I don't think that we'll make a whole lot of progress with tyrosine kinases going forward. It's going to be other targets. And I actually think that, you know, Mother Nature isn't done. You know, actually, we still don't know why Taxol is in the bark of the yew tree. Okay? It wasn't to keep away the spotted owl. Okay? And it wasn't because the yew tree thought a man would come by and take a bite off the bark, you know, and try to eat it. So why is Taxol in the bark and in the leaves and everything of the yew tree? I don't know. So there's a lot of things that we don't know, but why I'm saying that is that I think Mother Nature has figured out which are important targets for lethality, and so we still have a lot to learn.
Okay, that's interesting. Just to put a follow-up on that.
Mm-hmm.
One thing always amazed me about Taxol, I know it's a very complex molecule, but when you look at the microtubule and the bind of Taxol, the microtubule, you figure out why do you need something that's that complicated? If you just get to basically our limited knowledge, and here we have other people that have been thinking about microtubule, it really amazed me. Why couldn't you do the same job with a simpler molecule?
Yeah, and maybe you could, but you know, I showed you the kinomes, and I showed you how all of these drugs have multiple targets. To this day, nobody has found a credible second target for Taxol other than the microtubule. So I think that Mother Nature has tweaked it and tweaked it and tweaked it and tweaked it
very specific.
until it is really specific. You know, I mean, Mother Nature is the best chemist.
But I agree with you.
There's no doubt about
It's very
it.
specific to the microtubule, but it grows on the yew tree, right? Why
Right.
these connections are interesting?
Exactly, and there's many more compounds like that. So Mother Nature went after the microtubules in multiple ways. So when you see that, you say this must be important, and it is, obviously.
So with a lot of these TNIP drugs, yeah, the patients develop, or the cancer becomes resistant after 1 to 2 years. And there's particular mutations that take place in the cancer cell. So can you make some comments about personalized medicine and mutations in cancer? What do you see for the future of oncology in this direction?
Yeah, so I'm probably one of the few people in oncology that doesn't subscribe to the personalized medicine mantra. And I mean, I think it is, but not to the level that it's being promoted. Obviously, for example, in melanoma, we know that if you have a BRAF mutation, you will derive benefit from a BRAF inhibitor. You will develop resistance in 6 months, as you're noting. And that if you don't have the BRAF mutation, you derive no benefit. So in that sense, we can personalize that drug for melanoma. But if you have even colon cancer, and you have a BRAF mutation, which is very frequently found in colon cancer, BRAF antagonists have not found a niche in colon cancer. So it's just because you have the mutation doesn't mean that it's important or critical. You know, cancer cells, sure, I'll take that mutation, I'll use it to my advantage, but it not only has to be that it's using it to its advantage, it has to be that it is critical to its underpinnings, and that if you take it out from under them, they will fall. So mutations occur, and my bias is that that's what's key about Mother Nature. The reason we have mutations developing is that we try to make drugs so precise and so specific that what you end up with is a hand in a glove. And you've got a pocket, and you've got this drug that fits perfectly into it. At that point, any little change in that pocket will pop the drug out. And so you have a pocket that has 20 amino acids lining the pocket, and any change on those 20 to anything else practically will be enough so that the drug no longer fits. And in Gleevec, in the use of it in CML, the number of mutations are endless. I mean, there's a catalog, there's hundreds of mutations that have been described because it's such a precise fit that any little change can knock it out. So, in that sense, Mother Nature has been more clever. We really don't know the pocket that Taxol binds. We just know that it binds. And I was always jealous because I worked on drug resistance, and if you work on drug resistance, what you'd like to do is give a drug and get a mutation so you can know where the drug is binding and so forth and so on. It's almost impossible to get mutations to Taxol. I mean, I have put 10 years into this, and we ended up with 2 mutations to Taxol. And this is a lot of postdocs doing a lot of work on this and on other things as well. And yet, you take Gleevec, you put it in, and you get all sorts of mutations. So it's not only about specific, but how you make specific. And for us, making it in that way is not. But getting back to your question, so it needs to be the right target in the right cancer, and just because you have a drug for that target and you find it mutated in another cancer, it won't necessarily work. And so what bothers me is that these days, people are thinking, I'm going to go get my cancer, I'm going to go get it, analyzed genetically, I'm going to go with my doctor, he's going to reach in the shelf and said, this is the pill for you, go home and take it and live happily ever after. And unfortunately, that doesn't work that way. So, but obviously, the drugs that we develop, as in BRAF for melanoma, or EGFR for patients who have mutations in the EGFR inhibitors, we can personalize that. But I think we're so far away from personalizing the average patient that it's way too premature. But if you're a cancer center who doesn't offer that, patients aren't going to go. So everybody ends up offering it.
Just a quick question. You used the word network, I think, only once in your talk. And I wonder, are you or your colleagues looking much at metabolic reprogramming that takes place in cancer cells?
Yes, I mean, that's become a target. And actually, with regards to personalized medicine, my wife, who knows how much I dislike that term, sends me the other day an article that says, you'll like this. And it was depersonalized oncology. And it was targeting, basically, metabolism. Which is all the rage right now, to develop drugs targeting metabolism. Actually, I always tell the fellows that we train, they've been through high school, they've been through college, they've been through medical school. I said, nowadays in high school, you learn the Krebs cycle, and then you forget it. Then you go to college, right, and you learn the Krebs cycle. You all did, and then you forget it. Then they go to medical school and they again learn the Krebs cycle, and then they forget it. And then when they come, I said, you got to learn it again, because actually there's now 3 mutations in Krebs cycle enzymes that cause specific cancers. And actually, it's really interesting, getting back to what you were talking about. So there's mutations in isocitrate dehydrogenase, fumarate hydratase, and succinate dehydrogenase, both the alpha and the beta subunit. And if you have a mutation in succinate dehydrogenase, you'll get a very rare cancer, pheochromocytoma. And you might get some kidney cancer. This mutation, you inherited this, and it's in every cell in your body. But you get only these 2 cancers. Why? So every other cell in the body has it. What was it about the adrenal medulla, where pheochromocytomas originate, that it usurped this mutation to its advantage? So it's very interesting, but yeah, so metabolism is one of the hot areas now in cancer. Now, when you think about it, you say, well, every cell does metabolism. Now we're looking at really a lot of off-target effects and toxicity, and that may well be the case. But in these that have unique mutations involving the Krebs cycle, maybe there'll be drugs that can be developed for that. Okay.
How would allosteric effectors to open up the binding pocket for better inserts and so on?
Uh-huh. Interesting question. I think if it opened it and then closed it on the drug, it might work really well. Now, that would be really hard in a human because, you would end up having to give both drugs simultaneously, but I guess it could occur if it opened it and then the drug went in, it closed on it. Is that what you're talking about, for example? And then that was now an irreversible inhibitor, then that would work. And so it's an interesting concept. It'd be worth a try. And you might be able to develop a drug then that would be maybe more specific in some ways, but only after it's been able to be accommodated with a conformational change. Right. Right. But if you depended on the allosteric inhibitor for the drug to work, then you could increase the specificity of the drug. So that would take it to another level where you might say that it affects, 20 kinases in the kinome, and all of a sudden it's down to 1 or 2 only, and you've increased specificity. So, yeah. See, that's why everybody's got to work, or just half of you anyways. Initially, then eventually everybody. Right, Herbie? So, okay. All right, thank you. Thank you.